type of organic molecule we're going to go over is nucleic acids. Nucleic acids, the monomer is made up of a sugar group, a phosphate group, and nitrogen base group. Now the nitrogen base groups can vary. Especially, of course, in a polymer, you may have different nitrogen base groups, and so the monomer would be called different things. Polymers, of course, are made again through dehydration synthesis. Nucleic acids are carriers of things. They carry energy, they can carry electrons, or they can carry information. Energy carriers, a typical one is ATP, or adenosine triphosphate. That is, it has 3 phosphate groups on its sugar, nitrogen, other connector. ATP is the major energy carrier for all cells, where the breaking off of a phosphate group will deliver energy to a chemical system. And we will talk more about that in a little while. Electron carriers are, some examples are NADH and FADH2. There's some other ones too, and you're going to meet these when we talk about respiration. They are going to be carrying excited electrons from one location to another without them losing energy, which is an important feature. And then lastly, we can have information carriers. Typical ones are DNA and RNA, and in there, because they are polymers, they are carrying many different, or at least some different nitrogen bases on their groups, so they have different ones. And because of that, they can code for information. And we will talk a lot more about that later. But let's just look at DNA a little bit closer here. DNA is made up of what they call a sugar phosphate backbone. So this is the sugars and the phosphates that are joining the parts of the molecule that's making the polymer. What's hanging off that is not connected are the nitrogen base groups. So these nitrogen base groups, in this case, they are different because of their ring format, and the ring format is either has 2 parts or 1 part. Here is just a closer example. Now RNA has some slight differences than DNA. DNA would be a double helix, so this is right here, this is just one side of the molecule. A double helix would have a whole nother side to it, and I'll show you that in just a second. But in terms of these nitrogen base groups, these are the nitrogen bases. Pyrimidines excuse me, are cytosine, thymine, and uracil. Uracil exists in RNA instead of thymine. And then purines are adenine and guanine. They are the same in RNA and DNA. Now in the DNA molecule, there's 2 sides. So what's represented here in the blue and the yellow are the sugar phosphate backbone, and the orangey colors right here are the nitrogen base groups that are hanging off in the middle. And what would happen is that they would join via hydrogen bonding in the middle. Okay, now the one thing I wanted to kind of finish up with, anytime that we have hydrogen bonding, so like in this case, but also in the proteins, if you have hydrogen bonding and you add heat to that substance, so in this case it would be to DNA or nucleotide, nucleic acids, or in proteins, because remember sometimes they had hydrogen bonds. If you have hydrogen bonds, and really actually ionic bonds in some cases, you're going to denature them, or that is make them fall apart if you add heat to them. So this is why having a really high fever, for example, is bad, or why how you can cook food that has proteins in it because you're denaturing those proteins. Also, another thing to know that I wanted to just finish up with this organic molecule section is in the homeworks I gave you structures to recognize. So you need to be able to, you don't have to draw out the structures that I've been giving you, like the monomers and the polymers, but you should be able to recognize, say, a sugar versus a nucleic acid or a protein, and what the differences are. or I'm sorry, amino acids is what I meant to say, and what the differences are. Notice that nucleic acids, for example, contain a phosphate group, in addition to the sugar, which has carbon, hydrogens, and oxygen, and a nitrogen. All right, so if you were to look at a protein, what did it have that was different than this, or what was it lacking? So make sure that you are paying attention to that.